Hi everyone, welcome back to Class 12, Chapter 5 Principles of Inheritance and Variation. So, in this chapter, this is a part 3 video of this chapter. So, the rest of the two parts are covered based on these chapters. The link are provided in the description. You can go and check it out. So, this part is based on incomplete dominance, concept of dominance, codominance, and multiple alleles. Okay, so if you are new to our channel, kindly subscribe to our channel and click the bell icon for future notification and follow our channel and kindly like this video if you like it. Okay, so now let us quickly dive on to today's concept and look after all the concepts in detail. Okay. What is incomplete dominance? Incomplete dominance. See, initially we studied that only dominant character is seen in the offspring, right? In the monohybrid cross, if you just cross two plants in a monohybrid cross means only one character is involved. If you cross two plants, the F1 progeny that you get will be only dominant, having the dominant character, right? That only law of dominant states, right, given by Mendel, right? So, and not only in monohybrid cross, even in dihybrid cross, also we studied in our previous session. When we cross two different characters also, in your F1 progeny you get only the dominant character is expressed in the F1 progeny. So we studied this in our law of dominance also. Okay, till now this is what we studied. Then what do you mean by the topic incomplete dominance? Means here the dominant character is not completely expressed. That is called as incomplete dominance. Incomplete means it is not completely filled, right? In between it is. That is incomplete. Okay, so if teacher asks you, you will say, if you have, do you completed your classwork means you will say, no ma'am, I'm incomplete. If you have not written some part of the classwork, you will say, I'm incomplete. That means you have not completed some part of the classwork. Similarly, only here incomplete means, so completely dominant character is not expressed in the offspring. That is called as incomplete dominance. Okay. So it is a form of intermediate inheritance. Intermediate means in between. We say no, in between the characters. Okay, in between. Intermediate means in between. Inheritance in which one allele for a specific trait is not completely expressed over the paired alleles. So you are having both pair, right? You have the paired alleles, you have the dominant character and recessive character. But both the dominant character and recessive character are not completely expressed in the F1 generation. Okay, the intermediate character is expressed. This results in the third phenotype. So phenotype is what? It is nothing but the physical appearance. We studied about it in our previous session, right? It is the physical appearance. So first physical appearance is all about the dominant character. Second physical appearance is about the recessive character. The third physical appearance is what we get in our F1 generation. That is the intermediate between the dominant and recessive. Okay. So, in which the physical trait is a combination of phenotypes of both alleles. Okay. So, that third trait will be the combination of both dominant allele and recessive allele. That third allele will be the combination of the physical genotype or physical trait in the sense the physical phenotype. Okay, so of both the that is the third phenotype will be the combination of the dominant character and the recessive character. Okay, so now you understood, you got a idea on what is this actually. So when we see an example, you can clearly understand this concept. Okay, so here it is observed in the dog flower or antirrhinium or snapdragon. So these are the names in which these are the names of the flower plant where you can able to observe this incomplete dominance. This is an example given in your NCERT textbook. Okay, so when we explain this example, you can understand this. Okay, so here are, we are going to take the homozygous red is crossed with homozygous white. Here the intermediate character is observed. Okay, so this picture I will explain you later, but before that I have to explain you one written format. Okay, clearly observe here. I am going to draw one sketch. Okay, so I'm going to take capital R, capital R. This is nothing but red. This is my dominant character. That is red flower. I'm going to cross this red flower with white flower. White flower. Okay. So here white flower, you are going to take it as small r. So small r. This is your white flower. Okay. So I'm going to cross. These are nothing but my parents. So these are my parents. So parents will produce what? The parents will automatically produce gametes. So what is the gamete they are producing here? Capital R will be produced here. Small r will be produced. When these two fuse, you will be getting capital R, small r. But here it is not red color. It is pink color. Pink color. So here the capital R, small r represents the pink color. So this pink color is your F1 progeny. Your F1 progeny will be in pink color. This pink color indicates the what? Intermediate character. With pink color indicates the intermediate, mediate, intermediate character. Clear? Intermediate character. Now you just see here, you can able to see the white color flower here, right? You can able to see the white color flower here. This white color flower is recessive in type. This white color flower is recessive in type. So this white color flower is crossed with this red color flower. Okay, this red color flower is dominant in type. This red color flower is dominant in type. This red color flower is crossed with this white color flower, recessive type. Automatically what is produced? This pink color F1 progenies are produced. This pink color F1 progenies are produced. Clear everybody? Got that? Okay. Now what happens is, we just clearly observe here. Clearly observe here, I am going to draw the another one information. Okay. So see here, we know that our capital R and small r is nothing but the pink color. So I am going to cross it with again capital R, small r. What is this? This is the self-pollination, right? So doing the self-pollination, performing self-pollination, what you will get? You will get two different types of gametes. Capital R is formed, small r is formed. Here also two types of gametes. Capital R is formed, small r is formed. Clear? So now what will happen is, you are going to put the Punnett square. Already we studied what is Punnett square, why we are putting the Punnett square in our previous session. If you have not watched, the links are provided in the description. You can go and check it out. So Punnett square we are putting it, and you are having the male character and you are having the female character, right? So then your capital R, small r is placed here, capital R, small r is placed here. So what you will get here? Capital R, capital R, capital R, small r, capital R, small r, small r, small r. So now if I just go and if I have to write it, what is this phenotype will be? Here the phenotype will be, it will be red in color. Here it will be pink in color. Here it will be pink in color. Here it will be white in color. White in white in color. So the here, see, similarly like monohybrid cross, the phenotype, you have phenotypic ratio. So you have two types of ratio. What are the two types of ratios you have? One is phenotype, phenotypic ratio, phenotypic ratio. So I'm going to write it here. And you also have genotypic ratio. Genotypic ratio. Okay. What is the phenotypic ratio of monohybrid cross? It is 3:1. What is the genotypic ratio of monohybrid cross? It is 1 is to 2 is to 1. So here also the genotypic ratio is same as monohybrid cross. Here also it is 1 is to 2 is to 1 only. But the phenotypic ratio is not like the monohybrid cross, because you can able to see that phenotype is physical appearance. Here you are having two pink color, one red color, one white color. So here also you are having 1 is to 2 is to 1 ratio. So you have to remember that whenever it comes for the incomplete dominance, your phenotypic ratio and genotypic ratio both are same, and both will be equal to 1 is to 2 is to 1. Okay, both will be equal to 1 is to 2 is to 1. So this is how you have to understand about the incomplete dominance. Okay, so what is incomplete dominance? You are taking the recessive character of white color. It's observed in snapdragon flower, that is given in your example given in your NCERT textbook. You are taking the recessive plant and you are taking the dominant plant red in color. Okay, dominant plant red in color and recessive plant white in color. When you cross, your F1 progenies all will be in pink color. Just this basic explanation you understand. Okay, clear? Hope you got. Okay, with this. Now let us move to the next concept. Our next concept is about the concept of dominance. So it is also a quite important topic. So what is actually the concept of dominance means? See, in a diploid organism, there are two copies of each gene, that is a pair of alleles. What is a diploid organism? Diploid organism means organism that have 2n. 2n is n is a number of chromosome, right? We have 2n. Okay. So diploid organism means 2n. What is haploid organism? Just n. N is the haploid organism. So in case of diploid organism, there are two copies of genes. We already know, right? A pair of alleles are present. So for example, you take the trait tallness, you have T, it is a allele. You have small t, it's a allele. So you have a pair of genes, right? Everywhere you have a pair of genes. Now also we took red color. How we took red color? Like this. How we took our white color? Like this. So this is how we will take, right? So this is a pair of alleles. You know what all these concepts. These two alleles need not always be identical as in a heterozygote. Okay, these two alleles, they will be most probably, that is uh, probability, most of the times they will be identical only. But it is always not necessary to be identical. In some cases, they can be different. Okay, so one of them may be different due to some changes, due to some characteristics features. Because of that, some changes can happen in both of the genes. Okay, so both of the genes can be different in some cases, but most probably the genes will be identical. Okay, so sometimes it may be different. So example, the gene contains. So now we are taking the example of the gene producing enzyme. So now you take a normal allele. Normal allele means both are identical to each other. Identical means not, it is not that you always have small r, capital R, capital R. You can have heterozygous or homozygous. Both you can have. But identical means they both possess the same character. That is what. Okay. So here if you see means a normal allele. Normal allele is nothing but normal allele states that it will be always identical to each other. That is called as your normal allele. So always normal allele will be identical to each other. It produces the normal enzyme. Normal allele is going to produce the normal enzymes only. That is needed for the transformation of substrate S. So normal allele is producing normal enzyme, that is transforming the substrate S. Already this concept we have studied in our class 11, right? Class 11 biomolecules. You know how the substrate S is converted uh, by enzymes. Okay, one concept is there related to that. Okay, so now normal allele is transforming the substrate S with normal enzymes. So theoretically, the modified allele could be responsible. So the modified allele is responsible for the production of the normal or less efficient enzyme. Okay. So modified allele, what is modified allele? Modified allele is not identical allele. They can be responsible for normal or less efficient enzymes. Non-functional enzymes, no enzyme at all. Three conditions are there. You have to remember three conditions are present in case of modified allele. The modified allele, if it is present, there are three conditions. One is normal or less efficient enzymes it can produce. Non-functional enzymes it can produce, no enzyme at all. Okay, so here if you see means a modified allele is equal to, so always modified allele will be equivalent to unmodified allele. That is, allele present will be equal in number, but it will produce the same phenotype, trait, everything. It will be producing the same phenotype, same trait, everything. But okay, in transformation of his substrate. Okay, such equivalent allele pair is very common. So uh, actually when modified allele is equal to the unmodified allele, what happens? They possess the same phenotype, same trait. Okay, they possess everything same. In they also convert the substrate to substrate S in the same way. This is very common, but in some cases you can able to see that they will not be equal. Okay, so they will not be equal. In that cases only we can able to see some differences. See this unmodified allele, unmodified functioning allele. It is representing the original phenotype. Okay, unmodified is representing the original phenotype of an organism. So you can able to see here original phenotype is dominant allele. Okay, see unmodified unmodified allele is dominant. Remember like this. Unmodified allele is dominant. Modified allele is recessive. Okay, unmodified allele is dominant. Modified allele is recessive. See, original phenotype is unmodified, right? So it is dominant allele. Modified allele is generally recessive allele. Clear? So these are the important concepts you have to remember here. Not much. Easy only. You can easily remember these things here. Okay. So let us move to the next now. So in codominance, now we are going to see about the codominance. Okay. See, codominance. Already we studied about incomplete dominance, dominant character, okay, recessive character. What is this codominance? Codominance means it is representing the co means usually you will say both, right? Okay, some advertisements like that you might have heard. So there they will use co word co. Okay, co means both. Okay, so here in codominance, what happens? Uh, the F1 generation resembles both the parents. The F1 generation is going to resemble both the parents. Okay, so if you just see means both the parents means what? Who are the both the parents? Mother and father are the both the parents. It represents both the parents. So that is called as codominance. Okay, if codominance is F1 generation resembles both the parents. So here we are going to take the example of ABO blood group. Okay, ABO blood group. First I will try to explain this concept because this concept is very easy concept. I will try to explain this concept. Later we will read all these definitions. So then you can able to understand these things very easily. Okay. So first let us try to understand these things. Okay. So now let us see here. Okay. Uh, just to see, okay, I'll just explain it briefly, then we will read the points and let us try to understand this concept. Okay, okay, just to see here, what we already know is you are going to just take the example of ABO blood group type. Already we studied about the blood group. What are the different types of blood groups are available for us? A blood group, AB blood group, B blood group, and O blood group. So four types of blood groups are available: A, B, AB, O. Okay, while we are writing ABO blood group like that only you will write. Okay, ABO grouping system we are going to study. So now when you see means, see this ABO blood group is controlled by the gene called I. Okay, this ABO blood group, this ABO blood group is controlled by the gene called I. I is a gene. Now this I gene has three alleles. Okay. Now usually we study that genes are having two alleles, but this is an exception case where here you are having three alleles. Okay. One is A is IA is one of the allele, IB is one of the allele, and small I is one of the allele. So this I gene is containing three alleles: IA, IB, and I, out of which these IA and IB are the dominant allele. Okay, IA and IB are dominant allele, and I is the recessive allele. I is the recessive allele. Why IA and IB are dominant allele? It is dominant allele because these two alleles contains sugar. These two alleles contain sugar molecules present on the surface of the plasma membrane. So on the surface of the plasma membrane, what is present? Sugar molecules are present. So that these two are called as dominant alleles. And this I is a recessive allele because here you don't have any sugar. So here sugar is absent. Sugar is absent. Since sugar is absent, it is recessive in character. Clear? So sugar is absent in plasma membrane, so it is recessive in character. These two are dominant. Okay. So here you have more than one dominant character and one recessive character. Clear this one? Okay. So now when we talk about the ABO blood group, already we studied in our class 11. Some things we studied about it in class 11. So A blood group will have some antigen. What antigen will A blood group have? A blood group will have A antigen, right? B blood group B blood group will have B antigen. AB blood group will contain both A and, so both A and B are present here. Antigen. Here no antigen is present, absent. So that only this O is called as what? Universal. What is this O? O is, O blood group is universal donor, right? Your O blood group is what? O blood group is universal donor, right? So what is your AB blood group? Since it contains both A antigen and B antigen, it can get the blood from A blood group also, it can get the blood from B blood group also. And AB blood group having people can also give blood for these people. O can also give. So since all the people can give, these are called as universal, universal acceptor. Universal acceptor, right? So based on these informations, I hope you got an idea on the codominance. So based on these informations only, we are going to study about the codominance. This is what. So now let us move for the wordings and let us see this. Okay. Okay. You see, the very good example for codominance is the red blood cells. Okay. So what is this red blood cell? That is ABO grouping in human beings. So in human beings we have only ABO grouping. So that is what I told. ABO grouping is A blood group, B blood group, AB blood group, and O blood group. Four types of blood groups we have. Okay. So I told no previous slide, ABO blood group is controlled by the gene I. So the gene I is controlling the ABO blood group. The plasma membrane of the red blood cells contains sugar polymers that protrude from its surface. The kind of sugar controlled by the gene. So this sugar is controlled by the gene. So in the plasma membrane, this is a plasma membrane, one sugar molecule will protrude from the plasma membrane. this sugar molecule can be of A or B. Okay, the types are there. So if it is A type, it will be present in the ARBC. BRBC, B type sugar will be present. AB, in AB both A and B sugar type will be present. Okay, so the gene has 3 alleles. So you can able to see that. Okay. Next, the gene I has 3 alleles. Already I told. If you can't able to understand, in previous slide I told no IA. A should be at the top. Since I can't type here, it is like this. So A should be at top, IB, B should be at top, and I. Okay, like this it should be. Okay. The alleles IA and IB produce a slightly different form of sugar, while allele I does not produce any sugar. So allele I and IB are dominant character. So they produce sugar. I is not producing any sugar because it is a recessive character. Because human are diploid. Diploid, already you know the meaning of diploid. What is diploid? Diploid means 2n, right? Diploid means 2n. Each person possesses any two of the 3 IG. Okay. Diploid means only 2 characters can be observed, right? So 3 alleles are available. 3 alleles are available. Out of these 3 alleles, only 2 alleles will be represented in one organism. In one organism, you can only expect 2 alleles. So these are completely dominant over I. I told no, that is what. Okay. So now let us see this tabular column. The same tabular column is present in your NCERT textbook also. NCERT class 12 textbook. This tabular column is present, and this tabular column is very important. This tabular column is very important for you. Okay, reading this tabular column will help you to answer various types of questions. Now, see allele from parent 1, allele from parent 2, genotype of the offspring, blood types of the offspring. So if you see here, we have 4 types of columns as we saw earlier, right? So now we are going to fill these columns. Okay. So you see allele from parent 1. So here allele from parent 1 is going to be IA. Okay, IA. Allele from parent 2 is going to be also IA. Both the parent is going to give IA. Then what will be your genotype of the offspring? Genotype of the offspring will be IA, IA. The blood types of the offspring. What? See, IA, IA means what type of blood it will have. Both IA and IA are dominant character. So, dominant character will be expressed as the blood group type. Then A blood group will be expressed. So, the child will have blood group A. So, if parent 1 is going to have IB and parent 2 is also going to have IB, what is the genotype? IB, IB. Then what is the blood types of the offspring? The blood types of the offspring will be B. So they are going to have the blood type B. Okay. Next, allele from parent 1 is going to be same IA, but parent 2 is going to produce I. So what is the genotype? IA, I will be the genotype. But I told I is a recessive character, so it will not be expressed. So IA is a dominant character, so IA only will be expressed. So the blood group will be A. Then IB, here I is present. Then what happened? IBI will be there. So IBI means you can able to see that your B is the dominant character. So your blood group type will be B. Then IA and allele 2 parent 2 is going to produce IB. So you are going to have IA, IB. Both are dominant characters. So your blood group type is going to be AB. If parent 1 is going to produce IB, parent 2 is going to produce IA, both are dominant characters, so you will get IB, IA, right? So it is also going to produce AB blood group. Now parent 1 is also producing small I, parent 2 is also producing small I allele. Then you will be having small I, small I. Both are recessive characters, so you will be getting the O type of blood group. Clear? So this chart is really important. You have to remember. Easy only now. So you can remember it easily. Okay. Now going for the next concept. It is multiple alleles. Multiple alleles. Okay, so there are more than two. So alleles, if more than two alleles are present, three alleles governing the same character is called as multiple alleles. So now only we saw an example. What is that? ABO blood group. Okay, ABO blood group type. Blood group type, that is red blood cells, right? Red blood cells. So this is an example for multiple alleles. Because how many types? So gene I is containing how many types of alleles? Three types of alleles it is containing: IA, IB and I. Three types of alleles are present. So since more than two types of alleles are present, that is three alleles governing the same character. Okay, it is governing the same character of blood group only. So that it is called as what? Multiple alleles. Easy only now. What is multiple alleles? Okay. Thank you so much for watching this video. That's all for today's video, and we came to the end. So if you like our channel, please kindly like this video and kindly subscribe to our channel for future notifications and future videos. So that's all for today's video. I'll be upcoming with next few parts of this chapter. So thank you everyone. Bye.